previous classes?
Ma'am, actually the files and the PDFs are not getting opened in the
Now
resources.
it will be opening. Now it will be opening because yesterday actually some problem was there, some technical problem, and it was resolved, I think, by yesterday night only. So you will be able to open all the files, whatever the files are. Fine. So I have uploaded there the PPT also, the structure of a leaf, and the reason for uploading there is like, those are very, very important part for your 1 marker question as well as 2 marker question. So the difference between the palisade, the stomata, guards, to learn them. Fine. And the structure also of a leaf, anatomy of a leaf is important because what do? They will be asking you to label. So picture they will be maybe they will be giving you the picture and they will be asking you to write down the name of ABC, or maybe what do? They ask you to draw the picture and then to label chloroplast, to label mesophyll, to label the stomata. So anyways, in both the ways it can be asked. Still, Aaron. So today we'll be discussing about the
Good evening, ma'am.
experiment. Good evening, Aryan. So regarding the experiment we are going to discuss, and this experiment is again very, very important experiment. Most of the year they ask for it, and it is a very, very simple. But they do not ask you directly, like to explain the experiment. What do? Between the experiment, there are few things, like whatever they have used in that. From that point of thing, they keep on asking the question. So why we have used this? What happened if we use this? So like this, you should know the complete experiment and you should know why we are using that, right? So Zoha, Aryan, and Meher, turn on your video, please.
Ma'am, 1 minute, ma'am.
Fine.
I'll drink coffee.
Okay.
Good evening.
Soha, how are you? Good evening, Shubhansh. Zoha, how are you? Thank you. Good. So, okay, still they are joining. Let them join, and then I'll be explaining the experiment. So, I hope you all have revised the last topic, which was the anatomy of leaf, and whatever the structures we have discussed, whatever the parts we have discussed, you have learned it. Yesterday, the resources were not opening, but today you try, it will be opening. Okay. So, Abhigya, Shubhansh, Meher, turn on your video. Hurry up, children. I should not keep on asking you to do the same. You should do it by yourself. Okay. Shubhansh, hurry up.
Ma'am, I posted the message in the group. I'll not be able to download my
So,
camera for today.
okay. So, okay, still they are joining. Am I audible, children?
Yes,
Yes,
ma'am.
ma'am.
Okay, but I'm not able to see you actually all. Okay, never mind. So, we'll be discussing about a very good experiment, and it is a very, very useful experiment. It's about the carbon dioxide. So, the plants required what? They require the carbon dioxide. So, if they want to grow, if they want to live, they need a gas called as a carbon dioxide. So, to prove that, what kind of experiment we have to do? So, that experiment we will be discussing in the today's class. Is the screen visible, children?
Yes, ma'am.
Okay. So we will be discussing that. So experiment, name of the experiment is, CO2 is essential for photosynthesis. So for the plants, what do they need? They need the carbon dioxide. If this comes for the complete writing, how you will be writing? You will be writing with the aim, right? In the grade 6, 7, 8, 9, you might have made the files also, the bio file, chemistry file, physics file. So you should always start with writing the aim. Aim means the topic of what you're going to prove. So in the aim, what you should write? That carbon dioxide is necessary for photosynthesis. So the aim of the experiment is carbon dioxide is necessary for photosynthesis. Now, second thing, what we should write is about the materials required. So, what all the materials we need for doing this or for proving this experiment, those materials we will be writing in the next thing. So, what do we need is basically we need two potted plants. Okay, what do we need? We need two potted plants. We need two bell jars. We need a dish plate. Then we need the Vaseline and we need the KOH. What do you mean by KOH, children? What is the full form of KOH?
Potassium hydroxide.
Very good. So it stands for the potassium hydroxide. So these are the things which, or these are the materials which are required to prove the experiment. Now, I'll be.
Ma'am,
Just
can you
give
once
me
go
a
back to the
minute.
materials required I was writing?
Yeah,
Yes,
I'm
ma'am.
going back. I'm going back. Hmm. So the materials required are the two potted plants, two bell jars, a dish plate, the Vaseline, and the KOH. So these are the five things we'll be requiring for the experiment. Two potted plants, two bell jars, a dish plate, Vaseline, and the KOH. And this Vaseline is basically the normal Vaseline we are using, right? So in our day-to-day life, we are using the Vaseline, a jelly-like cream, right? A jelly-like petrol jelly-like thing. So the same Vaseline, we should use it for doing the experiment. Now, this is a setup of the experiment. Fine. So you can see the two pictures are there. This is the A1. Just a minute. So this is the A picture and this one is the B1. Now, inside this, you can see what are there. They are the potted plants. So what is present inside there are the potted plants. So one is here, the second one is not B1. Apart from that, you can see inside the A picture, a kind of Petri dish is present. What is Petri dish? Petri dish is a kind of a glass-like dish. Okay, so it's a kind of the glass-like dish. Now, what they have done is for the A experiment, for the A setup, they have taken the Petri dish and on that Petri dish, they have kept the KOH. Fine. So they have what? They have the KOH here. So this pink color structure you can see this is what a KOH. Fine. Small amount of KOH they have added on that glass. Whereas in the second setup, can you see the KOH or can you see any kind of dish there?
No, ma'am.
No, right? So in the second setup, no dish is there and no KOH is present. Whereas in the first A setup, the KOH is there and the dish is also present there. Now, what they have done after keeping this thing, after keeping the KOH in the A and after not keeping the KOH in the B, what they did is with the help of bell jar, this is the bell jar. So it is also a kind of the glass-like jar. So by using the bell jar, they have covered both the plants, both the potted plants they have covered. So this is the down part of the bell jar. Okay, so this is the down part of the bell jar. So by doing so, what happened now? The air can pass easily, right? So by covering with the well jar, the air can pass easily. So what they do is, by using the Vaseline, they seal it tightly. So they will be applying this Vaseline on the down part of it. So where the spotted plants are present and covering the glass jar, what they will be doing, they will be packing it tightly with the help of the Vaseline. So once they apply this Vaseline, what happened now? The air cannot move in and out of it. Understood? So the reason why they have used that Vaseline so that it does not allow the air to move in and out. Am I clear, children?
Yes, ma'am.
Yes, ma'am.
Now, for example, like suppose this is what this is the bell jar. Okay, I think the picture is not visible. Okay. This is suppose, for example, the bell jar. Okay, so here this is the down part of it. The open part is the down part, right? What I'll be doing, I'll be keeping this bell jar like this, right? Now here from here the air can pass easily. So what I will be doing on this part, I will be applying Vaseline. So as soon as I apply the Vaseline here, what happened? It become seal tight. Now neither the air can go out, neither the air can come inside it because it becomes sealed because of the Vaseline. So that is the reason the Vaseline is being used in this experiment. Children, turn on your video. Abhigya, Shayan, Shubhansh, Aryan, Aman, Siddhi. Understood, children, how the setup is? Yes, children, yes or no? Understood?
Yes,
Yes,
ma'am.
ma'am.
Yes.
Okay. Shayan, Abhigya, turn on your video. Shubhansh. I should not
My
keep on
video is
saying,
on.
children, that. But I cannot see you. I don't know why. Okay, fine. So, that is what actually is the setup. So two potted plants. In one potted plant, they have kept the KOH on the petri dish. Second potted plant does not have the KOH. Then both the potted plants they have covered with the help of the bell jar. And after covering those, what they have used the Vaseline to make it airtight or seal pack. Right? Now, I'll tell you in the experiment why they have used the KOH. What is the main feature of KOH? And this KOH related question comes every year. The reason, or I can say the function of KOH is that to absorb the CO2. So what the KOH will be doing? KOH will be absorbing the CO2. Now you tell me what happened. In the A setup, since the KOH is present, the amount of the CO2 which is present there, what happened to it? It will be absorbed by?
Potassium hydroxide.
Potassium also.
Correct. So it will be absorbed by the potassium hydroxide. And if all the amount of carbon dioxide is absorbed by the potassium hydroxide, will the plant will be able to survive?
No, ma'am.
No, ma'am.
No, no. So it will not be able to survive. Whereas in the B setup, since no KOH was there, what happens? No absorption of CO2 will be there, and easily the plant can use the CO2 and it can grow further. Fine. So this is the
One
experiment all
question.
about. Yes. So this is experiment all about. So they sometimes they give you the picture and they ask you to identify the difference between both the picture. Sometimes they ask you to write the function of KOH, which is to absorb the carbon dioxide. Sometimes they ask you to write the function of Vaseline. Vaseline is used to make the airtight setup. So this is how the question comes from this part. And if they ask you to explain the whole experiment, then it is simple that you have to mention about the two potted plants. So we need to take two potted plants. In the A setup, KOH should be there. In the B setup, no KOH should be there. Then in the third step, both the potted plants should be covered with the bell jar. And then once they are covered with the bell jar, we have to make them airtight with the help of the Vaseline. And we should watch it for the some hours. After some hours in the A setup, the plant will die because of the absence of CO2, whereas in the B setup, nothing will happen to the plant. So hence proved that CO2 is necessary for photosynthesis. So by doing this experiment, it can be easily be proven.
मैम एक बात पूछनी है मैम दोनों प्लांट्स को वाटर भी तो चाहिए होगा फोटोसिंथेसिस के लिए वो कैसे मिलेगा फिर
Correct, 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 correct. So already the plant has the water actually.
मैम फिर दोनों प्लांट्स
So this
एक
is
साथ
no,
कई बार
Shubhansh, this is not for the long term actually. This is not for the long term process. We are talking only for the short duration. For short duration also, the plant need the carbon dioxide because water anyhow it is available with them.
ओके मैम
Fine. Water is already available with them. So they can use maybe for a day or for the 2 days they can use and they can survive. But CO2 definitely they need it because now if this KOH is there, they cannot survive for the hour also because all the amount of CO2 they are absorbing it slowly and gradually and they will die. Okay, so this is our experiment and this is like very, very important experiment to go on with. Am I clear, children, now?
Yes, ma'am.
Yes, ma'am.
Okay. Why the Vaseline is being used in the experiment?
to make the jars airtight.
Shivansh, are you Zoha? Okay, Meher, why the KOH? What is the function of KOH in the experiment?
Ma'am, KOH absorbs all the carbon present in a container.
Carbon dioxide, okay? Carbon dioxide in the container, okay?
Okay, ma'am.
I'll be showing you the other experiment also. Or we'll go to the NCRT book. Okay, your NCRT book. Now here, just give me a minute, it is getting loaded. Okay, so this was the experiment I was talking about. Activity 6.2. It is given in the NCRT book with the activity 6.2. Now, you can see here, the first step, take two healthy potted plants, which are nearly the same size. Second, keep them in a dark room for 3 days. Third, now place each on a separate glass plate and place a watch of one of the plants. So between both the plants, with one plant only, we should keep the KOH. The potassium hydroxide is used to absorb the carbon dioxide. So very, very important. So this KOH is used to absorb the carbon dioxide. Now the next step, cover both plants with separate bell jars. So as you can see in the picture, both the potted plants are being covered by the bell jars. Now next one, use Vaseline to seal the bottom of the jar to the glass plates so that the setup is airtight. So what we should do, this setup we have to make it airtight with the help of the Vaseline. Then keep the plants in sunlight for about 2 hours. The next one, pluck a leaf from each plant and check the presence of starch in the above activity. So what happened? Does it show the same amount of starch? No. There will be difference because in this, the KOH has absorbed the amount of CO2, whereas in this, no absorption was there. So that is what we can conclude from the activity that carbon dioxide is important for the photosynthesis process. Now coming to this part, the activity 6.1. This activity usually every time they do not ask, but yes, we should know about it. In this activity 6.1, I'll show you the experiment actually. How to go ahead with this experiment. I hope the screen is visible, children.
Yes, ma'am.
And just note it down somewhere. This website, olabs.edu.in, olabs.edu.in. So this site is very, very important for your experiments. Okay, I'll just give you a brief about this site. This was released during the COVID time by the CBSE. So CBSE they have released during the COVID time. So since in the school the children were not coming and it was very tough to explain the experiment, That is why what they have come up, during the COVID time with a conclusion or with a solution that whatever experiments are there for the grade 9, 10, 11, 12, they have made it in the animated form. So all those things are done by the CBSE and still we are following this site. So here, if I talk about the grade 10, so you can see all the bio experiment, whatever are there for your grade 10 are given here. What you can do, as like we are, moving to the chapters, as we are moving to the topics, one by one, we will be covering the experiment also. So for photosynthesis, we have here one experiment called as the importance of light in photosynthesis. So already we have discussed just now, carbon dioxide is important for photosynthesis. Same way, the importance of light also we should know. Now, they have these different things. The second one is second one are the procedure. The third one, animation. So in the theory part, you will be studying like what it is all about, how the process happens, what is the equation, where it occurs, role of the color, factors affecting. So do you think so this theory is important for you anywhere? Is it important for you to study this theory?
Ma'am, the theory only will know, ma'am.
Correct, actually. So when you study the experiment, this theory is anyhow related to your chapter also, right? If I talk about photosynthesis, process of photosynthesis, everything is there in your NCRT book also. That is why if you learn or read from this, you will be getting a much better idea, right? So read this theory part. After the theory part, they have given the procedure. So in the procedure, they have explained you how to do the experiments and what all are the materials required. So you can see the first material required is the healthy potted plants. Second one, hot plate. Third one, forceps. Then iodine, alcohol, boiling tube, distilled water, Bunsen burner. So for doing this experiment, these are the materials required. Now, in the procedure, they have given the step by step what we should do it, how we should do it, right? So right now I am not going with this. I am going to go back through the video we will be learning. So in the animation, a video will be there of showing the whole complete experiment. So I'll play this, listen to the video carefully, and then we'll be having a discussion on the same.
Importance of light in photosynthesis. To do the experiment, we require a potted plant, boiling tube, iodine, petri dish, methylated spirit or alcohol, distilled water, forceps, Bunsen burner. Take a healthy potted plant and keep it for 2 to 3 days in a dark place. Take 2 uniform pieces of black paper. Select a healthy destarched leaf and cover a portion of the leaf on both sides.
So what we need to do, we need to take a potted plant and then we have to keep that potted plant in a dark place for the 2 to 3 days. After that, we have to take 2 strips of the black paper and then we need to cover the leaf. Not completely, but yes, the part of a leaf we need to cover it from both the sides, the upper side also and the lower side also.
By using these 2 uniform pieces of black paper. Now, place the potted plant in sunlight for the whole day. Pluck the covered leaf in the late afternoon.
So after 24 hours, once the plant got the light, you need to plug that leaf and in the lab you should carry that.
Remove the black paper from the leaf. Take a beaker containing distilled water, place it over a Bunsen burner and boil it. Put the experimental leaf into the boiling water and boil for 5 to 10 minutes or alcohol. Remove the black paper from the leaf. Take a beaker containing distilled water, place it over a Bunsen burner and boil it. Put the experimental leaf into the boiling water and boil for 5 to 10 minutes till the leaf becomes soft. Remove the beaker from the tripod stand and allow it to cool for some time. Take another beaker containing distilled water and place it over a Bunsen burner and boil it to about 60 degrees. Take the bottle containing alcohol and pour it into a clean boiling tube. Place the boiling tube into the beaker containing water that is boiling. Take the leaf from the beaker using a forceps. Place the leaf into the boiling tube containing alcohol.
Now, the reason actually, after boiling the leaf for some 5 to 10 minutes, the reason why now they are again boiling in the alcohol so as to remove the color. So which is called as the decolorization. So decoloration of leaf is done with the help of the alcohol. So again a one marker question, why they are using the alcohol in this experiment? So as to decolorize the color of the leaf. So whatever the color of the leaf is there, it has to decolorize. It means it has to remove from the surface of leaf. And that is the reason that again they are boiling it in the alcohol.
Take the leaf from the boiling tube using forceps. Dip the leaf in a beaker containing distilled water and wash it. Place the leaf in a petri dish. Take iodine solution using a dropper. Drop a few drops of iodine solution on the leaf. Observe.
So what happened like after taking out from the alcohol, when you put the iodine solution on it, you will be finding that the place where the black strip were there, it does not convert it into the blue black, whereas the remaining part of it gets converted into blue black. So it was due to the presence of starch. So blue black color appears because of the presence of starch, and in the area of the black strip, what happened? The light was not there, and due to that, the starch was absent at the place.
After iodine treatment, the color of the exposed portion of the leaf turns blue-black, while the unexposed portion of the leaf turns pale yellow. Conclusion: starch is one of the end products of photosynthesis and turns blue-black on contact with iodine. The exposed portion of the leaf that turns blue-black shows photosynthetic activity, while the unexposed portion shows the reverse. This indicates that light is essential for photosynthesis.
So I hope children you are clear with the activity, what we have to do in the activity. Fine. So we have to take a potted plant. After taking the potted plant, we have to keep it in the dark for almost 2 to 3 days. Then we have to take a black strip, both with the equal size, covering both the parts of the leaf, upper also and the lower also. And then we have to keep it for the sunlight for 24 hours. After that, remove that leaf from the potted plant, and then boil it in the water. Then once you boil it, add it into alcohol. Reason for adding into alcohol, so as to decolorize it. Once the decolorization happens, then what we should do? We should add the iodine solution. So when you add the iodine solution into that, you will be noticing that the part which was covered will not be turning into the blue black, and the part which was covered, it will not be turning the blue black. So hence it shows that light is important for the photosynthesis process. Clear? Abhigya, Shayan, Meher, Abhinav, Shubhansh,
Yes,
Siddhi,
ma'am.
Aryan,
Yes, ma'am.
Aman,
Yes, ma'am.
Riddhi, Zoha. So both experiments are very, very important for the exam point of view as well as for your board practicals. Fine. So CO2 experiment, how to prove that, and the light experiment, how to prove it, whether it is important for the photosynthesis or not. Yeah. Now again, I'll share the screen.
Ma'am, why did they boil the leaf in the distilled water in the beginning?
So as to remove the alcohol. so as to remove the alcohol. What happened if something, we boil in the water? So alcohol was there on the surface of the leaf. So that is the reason they have boiled again in the water so that the extra alcohol which was there, it can be removed. And before the experiment, why they wash it? To remove the dirt particles. So whatever the dirt particles are there, they can be removed out of it. That's the reason.
Okay, ma'am.
Okay. We'll go to the next part of this now. So this was the animation we have watched. After we have the simulator. So it's like you can try it by yourself. So what you can do, there will be somewhere, the So here, basically, it means you are giving them the instructions to work accordingly. So select the power source. So 40 watt or the 100 watt you need. So 40 watt is with the less light and the 100 watt is with the more power. Then select the distance of the power source. So how much distance you need? 50 centimeter is here, 100 centimeter is far. Fine. So according to your choice, the sources or the object will be moving. Now select the color of the filter. What color do you need? You need the red color, or you need the green color, you need the blue color, or you need the clear color of the light that you will be selecting. And then start. So once you start, the watch is moving. You can see the watch is, rounding. It is moving in the clockwise direction. So with the help of this, you will be able to know like how much time it will take to complete the whole process. So once you stop, there will be no risk because we have stopped it very soon. So idea what happened in the experiment. So here down you will be getting the idea of it. So that is what like you can try it at home by yourself by changing the centimeters, meters, by changing the color of the light, and you will be knowing in which color what happened to the source of it or to the object or to the plant. So simulator is for you to do it. You can do it. You can try doing it. Then in the Viva voice, basically the question which comes in the exams are there. So which one of the following is the correct definition of photosynthesis? So which is the correct answer for this? Question number 1. Correct definition of photosynthesis.
Ma'am, option 1.
Yeah, option 1. Conversion of light energy from sun into the chemical energy. Now this. Yes, Ayunija, Dhriti, Shubhansh, Abhigya,
Which question, ma'am?
Zoha, Aryan, Shayan. Question number 3.
Ma'am,
Which gas is
O2.
produced during
Ma'am,
photosynthesis?
O2.
O2.
O2. The answer for the fourth one, what is the importance of photosynthesis?
A part.
A part. So this process traps energy from sunlight and synthesizes food for all other living organisms. Okay, now question number 7. Why does a leaf to be tested for the presence of starch have to be boiled in alcohol? Why it has to be boiled in alcohol?
To dissolve
To
chlorophyll.
Yes, to dissolve chlorophyll. Okay, question number 9. Why should a potted plant be kept in the dark for at least 24 to 48 hours before performing the experiment?
Option 2, ma'am. To de-starch the
To
Yeah,
de
to
de-starch.
de-starch the leaf. Fine. So this is actually a very important, website which was released by CBSE, and not only for the bio, for the chemistry also, for the physics also. So this is actually the major screen of it. Okay. So grade class 10, you will be able to see now physics experiments here. And if you want, then more will be there for the chemistry as well as for the, yeah, here is for the bio. And then this is for the chemistry. So these are for the chemistry. Okay. So now whenever the teacher, whatever topics they are completing, it is your responsibility that you should check it. So if that topic is being completed, for example, the acid base has not been started, right? So ma'am might have started some reactions, chemical equations and reactions. So whether if you have completed the decomposition reaction, go for the video of it. Double displacement, go for the experiment of it. So this actually combination reaction, decomposition reaction, single displacement reaction, double displacement reaction is the part of the chemical equation reaction chapter. Okay. So be thorough with these experiments also because they are connected with your chapters as well. All right, children, I hope you all have noted down the website O Labs. Okay. Anything any one of you can ask me if you have any doubt.
No, ma'am.
And it's very, very interesting actually. The whole grade 10 is very interesting. So many experiments are there and so many activities are there to learn on. The only thing, whatever is done in a class, you should learn then and there. Don't keep on piling it for the, last, for the last to learn. Don't do that. Then and there, learn it. Fine. Today we did 2 experiments. Today itself, we should learn both experiments. And there is nothing to learn, you should only understand. Boiling should be done in distilled water. In the alcohol, it has to be boiled to decolorize the chlorophyll. Then again, a water wash should be there so as to remove the alcohol. Then iodine should be added to it. Then after adding iodine, the color will be changing. So all those things, see, nothing is there to learn, right? Only we have understand the different steps of it. Now it should be there with you. So understanding is very, very important for you all. I hope I'm clear, children. So you
Yes,
can
ma'am.
write in your own words now. Now you know what are the steps. You can write in your own words. There is nothing to mug up. It's very, very easy to learn. So, okay, learn just why it is needed, how it is needed, for what reason. So those why kind of things you should know. Why KOH is important, why the Vaseline is important, what is the function of KOH, what is the function of Vaseline, what is the function of alcohol. So these are the why kind of questions which comes every year. All right, children? And coming Sunday, you have the subjective test. This Sunday, I don't want you to give me the excuses, ma'am. I did not see this happen. I went out. No reasons will be accepted. No excuses will be accepted. Make sure that Saturday itself you attend the test paper. Saturday, 5 p.m. onwards, the paper will be live. You attend it and upload the answer sheet on the Wise app itself. No excuses will be accepted, children. Last time it was like first time, so we accepted it. But this is now the second time. We are not going to accept it. It's for all and everyone. And make sure, children, after every question, you should leave the line because it's very, very complicated, to check the answers if you have not left the line in between. So leave the line, write neatly because your presentation also matters a lot. Okay?
Okay.
So we'll meet you in the next class now for the next week. If any doubt you have, you can message me, okay, personally, and then clarify the same.
Thank you, ma'am.
Okay, ma'am. Bye.
Children,
Have a
thank
good
you,
day.
children. Thank you, thank you, thank you.